Yeah, all right. Thank you very much for your introduction. Hello, everyone. My name is Hossein. I actually graduated a couple months, maybe 3 months ago, from with my PhD from Dr. Alan Liu's lab at the University of Michigan. And it's my pleasure to share with you this work that I did during my PhD on design and computational analysis of biomolecular classifiers that work based on molecular sequestration and operate in an environment with shared resources. Biocomputation is one of the most important drivers of important physiological events, such as adaptive immunity, as well as cellular differentiation. During differentiation, we have stem cells that receive different inputs with different concentrations and intensities, and the cell essentially processes these signals and decides whether to what kind of, downstream cell type it wants to differentiate into. And the mechanism of cell signal processing usually relies on the receptors that reside on the cell membrane. These receptors, sense different inputs and basically transduce those inputs to the inside of the cell by activating signaling proteins. And the signaling proteins then interact with each other in a network-wise manner and basically translate the inputs to outputs, which oftentimes are in the form of transcriptional regulations in the cell. So inspired by this, what we really wanted to know and ask was that if we can create a small network with computational power for synthetic biological systems such as synthetic cells or engineered cells. And we wanted the small network to be the basic building block for even larger networks for more complex computations. And the approach that we took to answer this question relies in the special kind of a chemical reaction known as sequestration reaction, which I'm sure you heard about it a few times today. Sequestration reactions are biochemical reactions in which two species, say here the blue species and the green species, interact with each other and they form an inactive complex. So in other words, the blue species sequesters the green species. But if there's free green species in the reaction, it can form a dimer with the gray species and create an active output. Or an active dimer, which will be the output of our network. So if we have inputs, say X1 and X2, that can control the concentration of these two green and blue species, then we have a small network with interesting computational power, which I'll explain in the next few slides. So to represent this network, we chose sigma factors. Sigma factors are bacterial transcription factors that can form a complex with the core RNA polymerase in bacteria and drive expression of genes that are downstream of their corresponding promoters. Sigma factors also have antagonist molecules called anti-sigma proteins or molecules, and anti-sigma proteins essentially bind to sigma factors and make them inactive and unable to bind to core RNA polymerase. And the reason that we chose sigma factor and anti-sigma as our sort of representative molecules of the sequestration reaction is that we envisioned that we could have these, small signal processing modules in our synthetic biological systems, which have inputs that can regulate the concentration of a sigma factor, say S1, and anti-sigma molecule A1. And the output of this signal processing module would be the concentration of S1 complex with C denoted as C1. So to look at this system and model its behavior, we started from looking at the simplest form of interaction that happens in this system, which is molecular sequestration. So our model assumes that S1 is generated from X1 at the rate W1 and A1 is generated from X2 at the rate W2. And the sequestration happens at the rate gamma 1, and both species decay at the rate delta. Now, if the sequestration rate is fast, we get this behavior as the sort of the output of this sequestration reaction, which is the steady state amount of S1. And as you can see, this looks like a thresholding function where below a certain limit, we don't have any response. And above that, we have non-zero output. And that limit is essentially where there is more input, where we have more input that generates S1 versus the input that generates A1. And this behavior can also be described with this equation, which, although simple, it actually resembles this equation, which is the rectified linear unit or ReLU function, which is an activation function that's widely used in construction of perceptrons that are basic building blocks of neural networks. So because of this similarity, we called this output an activation function, and we called this unit a sigma-based perceptron. And the sort of the promise of a perceptron is that we know that a single perceptron can create linear decision boundaries where above or on one side of the decision boundary we have an output, and on the other side we have zero output. And the slope of decision boundary is tuned by the weights of the inputs to the perceptron. And if we put basically a network of these perceptron together, then we can have a nonlinear classification and computation. So basically what this means for us is that a single sigma-based perceptron can give us a binary classifier with a linear decision boundary. But if we make a network of these sigma-based perceptrons, then we can have nonlinear classification. However, in biochemical reactions, they have certain and unique physiological properties, such as resource constraints. For example, in a reaction, in a biochemical reaction, we might have a limited pool of resources, or we might have species competing against each other to bind or make complexes with this shared pool of resources. So to account for these kinds of features, we modified the model, and we considered the core RNA polymerase as the limited pool of resources that the perceptron will use. So we considered the binding of the sigma factor with the C, the core RNA polymerase that happens at the rate gamma 2, and we looked at the output of this perceptron as the concentration of the complex. Now, as you can see again, the activation function remains as a thresholding function, but here the output does not go to infinity anymore, and it rather saturates, the normalized value saturates at 1, which means that the maximum response that this perceptron can generate happens when it uses all its available resources. If you look at resource sharing, by simply adding another perceptron to the system that uses the same pool of limited resources, we get these activation functions, which essentially means if you have more of the competing perceptron using resources, for example, with having more alpha, which means more S2, which will use more of the shared resources, the thresholding behavior doesn't change, but this saturation limit actually lowers because there's less resources for S1 to use. And conversely, if you have more beta, which means more anti-sigma, then this saturation limit increases and gets closer to 1. And the implication that this sort of downscaling or suppression in the saturation limit of the activation function has is basically reveals itself if you look at the simplest architecture of network, which is made by only 2 perceptrons that are fed both by the same input. So if you look at the output of these individual perceptrons on their own, you can see that each of them have their own, linear decision boundaries. But if you look at their function in basically in conjunction, when they're both operating at the same time, you get these kinds of nonlinear behaviors, these sorts of perturbations that is imposed on these linear classifiers. Specifically, you can see them in regions where both perceptrons need to operate at the same time. For example, here, the perceptron number 2 takes most of the resources, and that shows itself as that suppression of the activation function in perceptron 1, and that causes this disturbance to the linear decision boundary. So we thought that how can we work around this problem, and we thought if this interference of these linear decision boundaries is the sort of the driving factor in these kinds of perturbations, what if we engineer certain network architectures in which the perceptrons in the same layer basically don't operate in the same input regimes, or their outputs don't interfere with each other. So we looked at a couple different examples, and I'm showing you one here just for the sake of time, which is a nonlinear classifier. So this network has 4 nodes, and what I'm showing you here is the output of the first 3 nodes in the first layer. And as you can see, each of these perceptrons demonstrate their own linear decision boundary. And these decision boundaries have little to minimal interference with each other. And this then allows us to add this second layer with the fourth perceptron, where we can have this nonlinear classification, sort of like a bandpass filter, where we have response generation only when the inputs are in sort of the middle range. Um, which is possible because the design was essentially made in a way that there is no interference between the outputs of the perceptrons in the first layer. So what I showed you today was that the design and analysis of sigma-based perceptrons. These are networks that work based on interaction of a sigma factor with its antagonist, and how putting together more of these sigma factor-based perceptrons allowed engineering nonlinear computation and classification. I should mention that this idea we've tested in vitro transcription translation systems, and we've been able to essentially demonstrate linear classification using sigma and anti-sigma molecules. Although basically the philosophy of this work was a forward engineering manner, where we wanted these systems to be used as input processing or biosensing modules. This also calls into question whether natural cells natively use these kinds of molecular interactions for signal processing and decision making. So with that, I like to finish my talk by thanking Christian, who I met at Cold Spring Harbor in summer 2022, my advisor, Alan, and people who helped with this project. And I'm happy to take any questions. Thank you.